So basically, what cloning means to make multiple identical copies of your gene of interest. That is called cloning. From a genome, you isolate your gene of interest and then you make multiple copies of it. That's basically cloning. Now this is of two types mainly, cell-based and cell-free method. Now this cell-free method is actually PCR, but in this video we will mainly focus on cell-based cloning. So let's understand the basic components of molecular cloning first. Number one, you need gene of interest, also called insert. Now this is the DNA sequence that you want to study, to clone, to modify, to express. Now either you can get it from genomic DNA or you can isolate RNA from your source and then using reverse transcription you can get cDNA. Then using gene specific primer you can amplify and isolate your gene of interest. Next you need is the cloning vector. That is, you need something, right, to carry your gene of interest into the host cell. So vector serves this purpose. The most commonly used vector is called plasmid, which are actually small circular DNA molecule. Scientists use it as a vehicle to carry your gene of interest into the host cell. Now a typical plasmid has origin of replication so that it can replicate independently inside this host cell. It has multiple cloning sites where your gene of interest will be inserted, and antibiotic resistance gene, which is extremely helpful for selecting the transformed bacteria. Next you need restriction enzymes, also called molecular scissors. Now these enzymes actually they recognize very specific DNA sequence and cut them and cut DNA at those sites. They cut both the gene and your plasmid using the same enzyme, and that will produce compatible end, which can be joined together with the help of DNA ligase. So this DNA ligase they form phosphodiester bond between your insert and this vector and will seal them together. So now that your recombinant plasmid is ready, it needs to be transferred to the host cell. So now you can transfer this to the host cell using electroporation. And now that you have a recombinant plasmid, after transformation, the bacteria divide rapidly, and every time the bacterial cell divides, the plasmid also replicates, as we see here. And as a result of which, millions of identical copies of your gene of interest will be produced. Next you need is a selectable marker as found in the cloning vector. Now why is it very important, this antibiotic resistance gene? So how do you know that every bacterial cell they received your recombinant plasmid? This is where your selectable marker comes in. Now most plasmid they actually contain an antibiotic resistance gene, right? Like let's say ampicillin resistance. Normally these bacteria they are actually very sensitive to this antibiotic. So if this bacteria they failed to receive this plasmid, they will die if plated on a media containing that particular antibiotic. But if those bacteria they carry your plasmid, they will be resistant to that particular antibiotic present in the plate where you have plated them. As a result, these bacteria they then survive and they form colonies. The selectable marker help us select the bacteria that have successfully taken up the plasmid. In other words, the gene of interest shows what you want to clone. The plasmid they act as the vehicle. These restriction enzymes they cut the gene and the plasmid, and DNA ligase they seal them together. E. coli or the host cell they get that recombinant plasmid and make millions of identical copies of them, and finally the selectable marker identify those bacteria that are actually carrying your recombinant plasmid. So now that you understand these 6 components, the actual cloning procedure becomes much easier to follow. So what is molecular cloning? Now, molecular cloning is the process of inserting a DNA fragment, that is your gene of interest, into a vector or plasmid so that it can be replicated inside a host cell like E. coli. The goal is to produce millions of copies of the gene or express the protein it encodes. Now there are many steps to it. Number one is gene isolation, where you either isolate genomic DNA or RNA from your source. Now in case if you isolate RNA, then make sure to convert it to cDNA using reverse transcription. The basic purpose is to obtain the DNA sequence that contain your gene of interest. Next is PCR amplification, where you design gene-specific primers. Then you amplify the gene using PCR and run the PCR product on an agarose gel. The reason is to produce millions of copies of only the desired gene and verify the correct product size. The next step is to excise the correct DNA band from the agarose gel and purify the DNA using a gel extraction kit. This is mainly done to remove primers, enzymes, nucleotides, and impurities before cloning. The next step is restriction digestion, where you digest both the purified PCR product and the plasmid vector using the same restriction enzyme. Now, the main purpose of this step is to generate compatible DNA ends that can join together. So basically, the next step is ligation, where you mix the digested insert and the vector with E4-DNA ligase and incubate under suitable conditions. So finally, you get your recombinant plasmid. Then comes transformation, where you introduce your recombinant plasmid into competent E. coli cells that is already prepared before. Steps used here mainly heat shock transformation or electroporation, depending on your lab manual. And then you recover the cells and plate them on selective agar. This is done to allow bacteria to take up and replicate the recombinant plasmid. Now select colonies that grow on antibiotic plates. Screen them using colony PCR or restriction digestion or blue-white screening. This is done to identify colonies that contain the correct recombinant plasmid. Now I have already made a video on my channel about colony PCR. If you want, you can check that out. The next step is plasmid isolation, where you grow a positive colony overnight. Then extract the plasmid DNA using your kit. The main purpose is to obtain purified recombinant plasmid DNA. Now, once it's done, then confirm the insert by restriction digestion and by DNA sequencing. This is done to verify the correct insert size, sequence, and orientation. Now, the main purpose of DNA sequencing here is to verify that the cloned DNA is exactly what you intended to clone. Restriction digestion tells you if the insert is present, if it's the right size, but it cannot tell you if the DNA sequence is correct. Sequencing confirms that correct DNA sequence is present, that is every nucleotide that matches the target gene. It will tell you if there is correct orientation, that is if the insert is in the desired direction, which is very important for expression. Sequencing also tells you that the insert is complete, that is no bases are missing or accidentally inserted. It ensures that there is correct reading frame, and it also makes sure that there is no PCR-induced mutation. For example, let's say if you expected a sequence like ATG, GAA, CCT, but in case you obtain ATG, GTA, CCT, so you can see that one base substitution, it could change an amino acid or even create a premature stop codon that will make your protein non-functional. So sequencing is the final quality control step. It confirms that your recombinant plasmid contains the correct gene with the correct sequence in the correct orientation before you proceed to downstream experiments such as protein expression, plant transformation, or functional studies.